In Chapter 9, we're going to be talking about the muscular system and learning how the muscles work, as well as the cellular histology part. First of all, the functions of muscle tissue. So there are 3 different types of muscle tissue, which each have their own functions. So we have skeletal muscle, we have smooth muscle, and we have cardiac muscle. And a lot of these are kind of intuitive in the name, but skeletal, this is what's actually allowing us to move. So it's responsible for locomotion or just movement, but also think about the movement of our face, our posture, some different respiratory movements, and other body movements. So things that are moving our body is part of the skeletal. So think about our bones or our skeletal system, it's moving those. And these are voluntary and controlled by the nervous system. So we have control over these, they don't automatically happen. The other 2 types, smooth and cardiac, though, are involuntarily controlled. So both the smooth and the cardiac are involuntarily controlled. So they're both regulated by hormones, which are part of the endocrine system, and still the nervous system, but the autonomic nervous system are kind of automatic. Again, that involuntary control. So then the cardiac, just to jump to it, again, the name implies it's part of our heart. So it's the movement of blood and then auto rhythmic. So it's the beating of our heart and the pumping of our blood. And that leaves us with the smooth muscle. So this is going to line the walls of our organs. So again, hollow organs, especially like our GI system, but also the blood vessels, our eyes, skins, and glands. Some of the functions would propel urine. So through the urinary tract, again, in the digestive, it's going to help mix the food and move food through the system, as well as dilating and constricting pupils and regulating that blood flow. In some locations, it is autorhythmic. All right, so just kind of a comparison here. We'll talk a little bit more about these as we move through, but just kind of starting out with the overall comparisons, and then again, we'll get into more details with each. So the skeletal muscle, again, it moves the skeleton because it's attached to bones. Its appearance, as we'll see, is striated, and it's the nuclei get pushed out here against the edges. So what it actually looks like under the microscope, again, we see these lines, they're called striations, with the nucleus kind of pushed out again to the border there. The smooth muscle is called that because it does not have striations. So you see here it's just nice and smooth. And then again, we kind of see the nuclei that are more kind of in the middle of these muscle cells. Again, lining the hollow organs, again, our blood vessels, eyes, and skin. And then finally, the cardiac is found in the heart. Again, we have striations. The difference here, how we can tell, is we get this branching here in the cardiac that we don't see. So in the skeletal muscle, it's nice and stacked. All of those are pretty much right on top, but we get this branching pattern that you can see here in the cardiac. All right, as far as some of their shapes, again, that skeletal muscle, long and cylindrical. The smooth is pretty similar, that kind of spindled shape, whereas again, the cardiac muscle is cylindrical, but it's branched is what makes it rather unique. And again, the nuclei, as I mentioned in the last slide, with the nuclei and skeletal, we have lots of them that are on the periphery or out on the edges. Smooth muscle, we just have one, and it's more in the center. Cardiac, again, also one, but it's centrally located as well. So as far as cell-to-cell attachments, there are none in the skeletal system. But again, we do have striations. Smooth muscles, there are gap junctions that join the smooth muscles together so that they can increase that communication. But again, they're smooth because they don't have the striations. And then the cardiac also have some connections. They're called intercalated discs that join them together. And then it's missing the information here, but they are striated. Just write that in. So yes, there is striations of our cardiac muscle. And then finally, some of the controls, again, as we already mentioned, the difference here, skeletal is voluntarily controlled. Now there are reflexes though, whenever we jerk or like the knee tap reflexes, those are involuntary. But for the majority, most of our skeletal muscle is voluntarily controlled, whereas the smooth is involuntarily. Spontaneous contractions kind of goes right along, same thing. So no, muscle cells don't spontaneously contract. Even those reflexes are not what we consider spontaneous contraction. However, both the smooth and cardiac do. And then the overall function, again, moving the body. Whereas with the smooth, we're moving that food through the digestive system, again, moving our urine, talking about vasoconstriction or dilation of our blood vessels, pupil size, and then contracting our glands, moving hairs, etc. And then the cardiac, again, in the blood or in the heart. So pumping the blood as well as contracting the heart. So moving all that blood through the heart, through the atrium and the ventricles. All right, so then our overall function, so each of those have their own, each tissue has their own function, but the overall functions of the muscular system, again, moving the body, our posture, breathing, also producing heat may not be one that we think about as far as our muscular system, but as we're shivering, again, that releases heat, which is one of the movements of our muscular system. Communication that allows for, again, the body to talk to each other, constricting those organs and vessels, thinking about like blood vessels, but also the GI system, and of course the cardiac, which helps contract that heart. Now the general properties of muscle tissue. So muscle tissue can contract. Again, that's something we would normally think about, and really contraction just means the muscle can shorten with force. We'll talk about how exactly that happens. It's excitable. Just meaning that it can respond to stimuli, which is typically nerves, whether it's the autonomic nervous system or not. Again, neurotransmitters get released from the nervous system, and it activates that muscle tissue. It can extend, so extensibility, so it can be stretched. So contraction is the shortening, extension is extending or stretching beyond its normal length. And even in that position, it can still contract. And it's elastic. So once it's stretched and extended, it can recoil to that original position after it's been stretched. So now if we look at the skeletal muscle anatomy here, so back to our connective tissues. So they're covered with this connective tissue, a few different types. So we have the epimysium. So this is the connective tissue that goes around the whole muscle, and we're going to be breaking all the parts of the muscle down. So the whole muscle is surrounded by this connective tissue called the epimysium. So again, like think about epithelial, it's on the outside. So the outside of the muscle. It does merge with the muscular fascia, which is kind of that little thicker layer of connective tissue that's adjacent to the muscles, and it's between the muscles and the skin. So it's a white kind of connective tissue that we get over top of our muscles. We also have paramecium. So this is some loose connective tissue. This is going to be around groups of muscle fibers. So we have the whole muscle made up individually of these muscle fibers within that. So each muscle fiber is covered by a layer called the paramecium. And then we have the endomysium, again, loose connective tissue. This is individual muscle fibers are going to be separated by this within each of those fascicles. And again, fascicles are just bundles of muscle cells that come together. And then we also have some collagen from that connective tissue that merge to form those tendons. And then that allows the muscle to attach to the bone. All right, so then the nerves and the blood vessels, so motor neurons, those are going to stimulate the muscle contractions. And again, that voluntary control. And each motor neuron controls several muscle fibers. So our neuron would be coming down, but then it branches at those axon terminals. So we have lots of branches coming off, and each of them could then be attached to like a different muscle fiber that's moving through. So it one neuron can control multiple muscle fibers. And each of those, again, has its own branch of that motor neuron. And then arteries and veins extend within the nerves through those connective layers we just mentioned. And lots of capillary beds for that gas exchange to provide oxygen to our muscles, as well as get rid of the carbon dioxide around each of those muscle fibers. All right, so now looking at the structure of what the skeletal muscle looks like. So the whole entire thing here is our muscle, whole muscle. And again, we're focusing on the skeletal muscle. So each of these whole muscles are made up of these individual bundles here, and these bundles are called fascicles. Okay, so we have our epimysium. Epimysium, that is going to be, get rid of this a little bit. So that epimysium, this is shown here in blue, that's going to be that connective tissue that completely surrounds the whole muscle. So then inside we have these individual circles here. These are going to be the fascicles, and the fascicles are lined and connected by the perimysium. And then we also have the muscle fibers, or the myofibrils, that are all the way inside. So we have a collection of the muscle fibers. These bundles come together to make up vascicles, and then the vascicles come together here to make up the whole muscle. And those individual units surrounding the muscle fibers, that connective tissue is the endomysium. Continuing with our skeletal muscle, which is where most of this chapter is going to focus on. So this develops from the fusion of what are called myoblasts. So these result in these, again, large multinucleated muscle cells. Again, one of the differences between skeletal and then the smooth and cardiac is that the skeletal muscle has multiple nuclei, and that's because all of these muscle cells are fusing together to make one big muscle cell. Again, it does have this striated appearance. That's due to some actin and myosin. We'll come back to that. So then the number of fibers remains constant, though after birth. So we can get muscles get a little larger, but that's due to hypertrophy of the muscle fiber. So the muscle fiber just getting bigger as we kind of exercise or work out. But the number of actual fibers remains the same, but they can increase in size. So the histology, what those muscle fibers, again, the muscle fibers are the smaller circles inside the myofibrils. Myofibrils combine to make the whole skeletal muscle. So these are the electrical component structures. These are what actually respond and transmit signals. So they have the sarcolemia, which is their plasma membrane. So again, because they're fusing together, the name of that is called the sarcolemia. And then the fluid inside is the sarcoplasm. And this is, again, just because we're talking about the muscle cells, but also because they're fusing. We have lots of cells fusing together. So again, the overall plasma membrane is the sarcolemma, the fluid inside the sarcoplasma. And then we have these T-tubules or transverse tubules. These are the folds of the sarcolemia. So that plasma membrane isn't just straight, so it kind of folds inwards and projects to the interior of the muscle. And we have the sarcoplasmic reticulum, or the SR. This is part of the smooth endoplasmic reticulum. So back to our cell components in our organelles, we have the rough ER and we have the smooth ER. Well, part of the smooth ER, especially in our muscle cells, the sarcoplasmic reticulum, this is where calcium is stored. And as we'll see in a little bit, calcium is what triggers muscle contraction. So the storage compartment is in the sarcoplasmic reticulum. And then we have some enlarged portions of that sarcoplasmic reticulum called the terminal cisterna. Those line those T-tubules, those folds of the plasma membrane of the muscle. And then two terminal cisterns, and their associated T-tubules, those are called a triad. So we'll see a picture here in a second. But again, we have these two terminal cisternae, and then the T-tubule that they're attached to. And here's what that actually looks like. So again, this triad here, we have the transverse tubule that goes through here, shown in green. So again, it literally transverses the entire muscle fiber. And then we have these terminal cisternae on each side of that transverse tubule. So those come together to make up the triad. So we have the transverse tubules and the terminal cisternae. All right, now looking at the histology of the muscle fibers. So this is kind of the smallest unit of that whole muscle. So the individual protein filaments come together to make up the myofibrils. So these are collections of filaments, and there are only two filaments. So we have actin filaments, which are thinner, and myosin filaments, which are thicker. And so those two myofilaments, typically just called actin and myosin, they come together to make up the myofibrils. And then those are inside of the muscle fibers, which are inside of those fascicles. And those myofilaments, the actin and myosin, form units called sarcomeres. Now we can kind of see the individual parts here of our whole muscle. So starting at the top, again, our whole muscle here covered by that epimysium shown in blue. This makes up the whole muscle. We have our tendon here that connects the muscle to the bone. So then the bone, or that muscle, whole muscle, is made up of these individual bundles here. Those are called the muscle fascicles. So we have lots of those tubules here that come together to form the whole muscle. Each of those fascicles are made up of individual bundles of muscle fibers. And then, so in what holds them together is that paramecium in between all of those muscle fibers. And then the muscle fibers are individually made up of myofibrils, which are collections of actin and myosin filaments, or myofilaments. Again, our actin are thin, our myosin is thicker, and those are going to form those structural units called sarcomeres. Now, looking at the individual functional units of our muscle, the sarcomeres, so again, this is the basic functional unit of those muscle fibers. So they have what's called the Z disc. That's the filamentous network of proteins. So again, those actin and the actin myofilaments are going to attach to those. And then it creates some regions called the I bands. So these are lighter staining regions containing the Z disc. And they also extend, though, to the ends of the myosin. So again, the Z disc attach to the actin, the I bands extend on to the myosin. The A bands are darker, so again, that creates the striations, this light dark pattern. These overlap with the actin and myosin myofilaments, except for in the center, but they overlap there. Then we have the H zone, which is the region in the A band where the actin and myosin do not overlap. So the A band is the overlapping region. H is where they don't overlap. And then finally, we have what's called the M line, which is in the middle of that H zone, again, not overlapping. And this is where those delicate filaments are holding the myosin in place. So in the muscle fibers, those A and I bands, again, the lighter and the darker bands, are in parallel of the myofibrils, and that's what gives that striated appearance that we have in those sarcomeres. We also have titin filaments, which are elastic chains of amino acids. This is what allows them to extend and be elastic. So if we look at our individual myofibril, again, these combined into bundles called the muscle fascicles. So if we just look, though, at the individual myofibrils, so we have the A bands and the I bands. This makes up the striations. The I band, the lighter region, is just the Z disc that attaches to the thin actin filaments. The darker bands, the A bands, if we look down here below, here in this A band, this is where the actin and myosin overlap with each other. So it makes it a thicker, so it's a darker structure. Again, our myosin is much thicker. The actin filaments, they're going to be a little bit thinner. But again, our muscle units, these sarcomeres, are made up of these A and I bands. Again, the I band just has the Z disc attached to the actin filaments, where the actin and myosin overlap is the A band. But there is a region inside of that A band, that, again, that darker region, where it's only the myosin. So the actin and myosin don't overlap. We just have a region of myosin, and that's called the H zone. And that H zone has the M line, which is just attached to the myosin. So again, our Z disc is attached to the individual actin filaments. the M line just has the myosin. And that's part of the M line, though, is part of the A band where we get the overlapping actin and myosin. But there's a little region in the middle where there's no overlapping, and that's called the H zone. So a little bit more about these myofilaments. The actin or thin filaments, they have two strands of this fibrous actin that twist around each other to form a double helix that just extends in length. And they again attach at either end of that sarcomere at that Z disc. It's composed of what's called G-actin, which is the monomer that builds them. And that active site can bind myosin during that muscle contraction that we'll see in a minute. There's also tropomyosin. This is the elongated protein that winds along the grooves of the F-actin double helix. So we have, again, the double helix just kind of wrapped around each other, but then a little elongated region called the tropomyosin. And then the troponin is made up of three subunits. One is going to bind to the actin. The second binds to that tropomyosin. And then the third binds to calcium ions. And this is space between the ends of the tropomyosin molecules in the groove between those F-actin strands. So then what we end up getting are tropomyosin troponin complexes that regulate the interaction between the G-actin and the myosin. Now the myosin, the thicker of the myofilaments, so this elongated myosin is shaped kind of like golf clubs. So it has the myosin heavy chain that's wound around to form kind of what's referred to as the rod portion. That lies parallel to the myosin myofilaments. And then there are two myosin heads that extend laterally. And those heads have active sites on the actin filaments that form what are called cross bridges. And they're attached to the rod portion by what's called a hinge region that can bend and straighten during contraction. So as we move through the steps of contraction, we'll see this a little bit more. There are also ATPase enzymes, and these are breaking down ATP to release energy needed for the contraction. And part of that energy is used to bend the hinge region of the myosin during the contraction. Looking now at what's called the neuromuscular junction, which as the name implies is where neurons join with the muscles. So those motor neurons that are part of the nervous system, they have electrical signals called action potentials. That's what allow neurons to communicate with each other and to whatever they're attached to. So those motor neurons have action potentials, and they stimulate muscle fiber action potentials that cause the muscle contraction. So that electrical signal from the motor neuron ultimately allows contraction of the muscle. So the points of contact, again, where the neuron meets up with the muscle fiber, is called the neuromuscular junction. This is also a synapse. It's just a specific synapse where we have a neuron joining with a muscle. So the presynaptic terminal, so before the synapse or before that junction, that's going to be on the axon of the motor neuron. So it has what are called synaptic vesicles, and those vesicles have neurotransmitters. And the neurotransmitter specifically involved in skeletal muscle contraction is called acetylcholine. So these motor neurons release acetylcholine into that junction, into that synapse, and then the neurotransmitters from one cell then can bind to ligand-gated ion channels on the next cell, in this case, a muscle fiber. So that is going to open up these ion channels that will ultimately lead to the contraction of the muscle. Now, the space in between the motor neuron terminal and the actual muscle is referred to as the synaptic cleft. And then the muscle, what's on the other side of this synapse, is called the postsynaptic membrane. So that's going to be the muscle cell, which is referred to as the motor end plate. So that's where that neuromuscular junction occurs. All right, so now looking at a few of the things we had just mentioned. So we've already covered, everything that's over here on the left-hand side, the parts of that sarcomere where we have our A bands and our I bands that cause those striations. But now if we look at what we just talked about, this neuromuscular junction is right here. So here's our motor neuron coming down. It has a nerve terminal, kind of like a little bud. Inside of that nerve terminal are the synaptic vesicles, and they are containing that acetylcholine, the little yellow dots. So whenever an action potential comes down the motor neuron, it causes the release of that acetylcholine into this space between the neuron and the muscle. And that is referred to as the neuromuscular junction. So just to kind of highlight here. So this region in here, all the way through there, that is the neuromuscular junction. Rid a little bit of that. So what's going to happen is whenever that acetylcholine is released from the synapse, it's going to bind to ligand-gated ion channels on what's called the motor plate. That's part of the muscle tissue where that neuron is going to be associated with. So at the other side of that neuromuscular junction. So again, just the other terms. So this is our presynaptic side, which is the motor neuron, and then the muscle tissue is the postsynaptic portion, which is that motor plate. All right, so then back to what we talked about on the previous slide. So again, part of our actin, we have the F actin molecules that are here. We have the tropomyosin, we have the troponin, and then those active sites here that are going to allow it to attach to the myosin. So again, just kind of looking at this, again, the tropomyosin forms this kind of double helix with those two that kind of wrap around each other to form that twisty tube there. And then that troponin, though, is going to allow it to bind to calcium, but it also binds to that tropomyosin. So it allows that interaction to occur for those individual actin molecules. And each actin molecule or active site has these those active sites are on that G-actin molecules that it's going to allow them to attach together and form that tropomyosin. Right, so muscle contraction occurs by what's called the sliding filament model, where literally actin filaments slide over myosins to shorten that sarcomere. So if we think about it again, our Z disc are attached to the actin. We have that M line where the myosin is. The actin and myosin, especially there in the A band where they're overlapping, they literally slide across each other. So if you take your hands and put one over top of the other and you slide them over top, that shortens the distance between your wrists. So that is that shortening of the sarcomere. Now during that, neither of those fibers change length. So again, if you're sliding your hands across each other and your wrists are coming together, your hands didn't shorten. They're not any shorter. Your fingers aren't shorter. They're just bringing your wrists closer together. So the shortening, though, is responsible for that contraction. Then during relaxation, the sarcomeres lengthen because of some external forces, again, like the contraction of antagonistic muscles. So muscles on the other sides contract and that pulls them apart. So they produce opposite effects. This is a really wordy slide, which I don't love, but it's good to have all the information kind of in one spot. So again, just looking at how these sarcomeres shorten, which is what causes muscle contraction. So starting with the relaxed muscle, which we have here at the top. So again, we've been over our Z disc here is attached to those actin filaments. They make up the I band. We just have the actin where we get overlapping of the actin and myosin. That's the A band. And then we have this area in the middle of the A band called the H zone. That's where we have this M line that's attached just to the myosin. So the Z disc attached just to actin. That M line is attached just to the myosin. Okay, so this is the relaxed state. Now what happens during muscle contraction is essentially we just get rid of that H zone. So there is no longer an area in the middle of that A band where the actin and myosin aren't overlapping. So they literally slide toward each other. So from each I band or each Z disc, that's going to shorten. So as the actin moves over or slides over the myosin, we get this alternating sliding, one going in one direction and one going in the opposite direction. So again, they literally slide across each other so that at this point during that muscle contraction, again, that M line has shortened those myosin interactions. And so we no longer have this H zone. There's no longer a region where we don't get the overlapping of those actin and myosins. Again, the actin and myosin fibers didn't get any longer or shorter. They just slid across each other more, which brought these Z discs closer together than what we have up here at the top. Again, and that's just because we're getting rid of that H zone where there is no overlapping. And that's what leads to that muscle contraction. So then as we move through the rest of this chapter, we'll see how that all works together to finish that contraction.